All right, everyone, get ready for a deep dive. Today we're tackling gene mutation.
Oh, interesting.
Yeah, we're going to be using this source. It's called CH15 summary gene mutation.pdf.
Okay.
And honestly, it's pretty amazing stuff. We're talking about these tiny changes in our DNA that have these huge effects. I mean, from evolution to diseases to even cancer.
Yeah, that's a big topic.
Yeah. You ever heard about those mutation hotspots, by the way?
I have heard of those.
Or that your dad, is probably contributing more new mutations than your mom.
I have heard of that.
Yeah, it's some wild stuff. But let's start with the basics,
Yeah.
What exactly are we talking about when we say mutation?
Well, at their most basic level, mutations are just changes.
Yeah.
Changes in the DNA sequence. It's like, think of DNA as your instruction manual, right?
Right.
Like for building an organism.
Okay.
So a mutation is like a typo or an edit to that manual.
So, are all mutations, I guess, created equal then? I mean, some typos are worse than others.
Exactly, yeah.
Yeah.
Not all mutations are the same. They can be small, like just one letter change, or big, like whole chunks of DNA getting moved around. And where these mutations happen in our cells, that really matters too.
Oh, right, right. The source talked about germ cells and somatic cells.
Yeah.
What's the difference?
Well, germ cells are those involved in, reproduction. So that's the sperm and egg.
Okay.
Mutations in these cells can get passed on to your kids, right? So they're driving evolution, and they can cause those inherited diseases we hear about.
So like a recipe being passed down through generations, but each time, maybe someone adds a little tweak.
Exactly. But somatic cells, those are the rest of the cells in your body.
Oh, okay.
And mutations there, they're more like personal edits to your own copy of the instructions. They might cause things like cancer, or even create little patches of unique cells within you.
Like a genetic birthmark almost.
Yeah, that's a great way to think about it. Now, let's dive into the different types of mutations that can happen.
The source, it mentioned a bunch of them. Is there a way to group them to make sense of it all?
Oh, definitely. One way is to look at the molecular change involved. For example, point mutations.
Right.
They're like typos in the DNA sequence, but they have different effects too. Like you've got missense, nonsense.
Wait, hold on. I'm a little fuzzy on missense versus nonsense.
Okay, so a missense mutation, think of it like changing one word in a sentence. It might change the meaning, right?
Okay.
But a nonsense mutation is like putting a period in too early. You cut the sentence short.
So nonsense is usually worse then.
Yeah, often. Then there are those silent mutations.
Right.
Those are sneaky because they don't change the amino acid in the protein.
But they're still mutations.
They are.
Or.
And they can still have subtle effects.
Yeah.
Like how much a gene gets expressed or even the shape of the final protein.
Wow. It's wild how even small changes can have an impact, huh? The source also mentioned insertions and deletions.
Right.
How are those different from point mutations?
So insertions and deletions are about adding or removing nucleotides.
Okay.
They can be one nucleotide or a whole bunch. And when these changes aren't a multiple of 3, it really messes things up.
How so?
Well, it's just the reading frame of the gene. Imagine reading a sentence, but every third letter is gone.
Oh, yeah. That'd be impossible.
Exactly. Same with genes. The protein it makes ends up totally different, often useless. That's a frame shift mutation.
Yikes. So we've got mutations by molecular change, but the source also groups them by how they affect the gene's function.
Yep. We've got loss of function where the gene doesn't work as well. Those are usually recessive.
So you need 2 bad copies for the problem to show up.
Right. But there's this twist. Dominant negative mutations. Even one bad copy messes up the good one.
Like a bad apple spoiling the bunch.
Kinda. Then there's gain of function. The gene gets a new or stronger ability, and usually just one copy is enough to see the effect.
So loss of function is like taking something away, and gain of function is adding something.
Perfect analogy. And lastly, we have suppressor mutations.
Those sound cool.
They are. A second mutation actually fixes the problem caused by the first one. It's like nature's way of correcting its own mistake.
2 wrongs making a right, genetically speaking. So where do all these mutations come from in the first place?
Well, some just happen naturally. DNA replication isn't perfect.
Wait, our cells make mistakes copying DNA?
Even the best machinery has its limits. There's a chance for errors, just like a typo. And things like tautomeric shifts can happen.
Botato?
Tautomeric shifts. It's like a word having 2 pronunciations, same letters, but different outcome. Basically, the DNA base temporarily changes shape, causing it to pair incorrectly.
Ah, okay. So it's
Okay.
not all perfect in there. And the source said mutation rates aren't equal everywhere, right?
That's right. Different organisms, different genes, they have different rates. Why? We're still figuring that out.
And something about a dad's age mattering too.
Oh, yeah, big time. It's because sperm cells keep dividing throughout a man's life. More divisions, more chances for errors to sneak in.
So an older dad means,
Potentially more mutations passed on. But don't worry, most of these are harmless. On average, you probably have about 60 new mutations compared to your parents.
wow, 60. It's like we're all unique snowflakes, genetically speaking. But what about those mutations that aren't just random accidents?
Ah, those are induced mutations caused by something outside damaging the DNA.
Like radiation, stuff like that.
Exactly. Certain chemicals, UV light from the sun, ionizing radiation like X-rays, they can all mess with DNA and cause mutations. It's why smoking is linked to cancer, right?
Yeah.
The chemicals in smoke are powerful mutagens.
And why sunscreen is our friend.
Absolutely. UV damage can lead to skin cancer. Interesting thing is, while we worry about human-made radiation, a lot of our exposure is actually natural from cosmic rays and stuff in the Earth.
So we're always under attack, kinda, both from inside and out.
It's true, but the good news is our bodies have developed ways to fight back.
Oh, good. Our cells can fix these mutations.
Yep. We've got these amazing DNA repair mechanisms. They're like our internal repair crew, constantly working to keep things in order.
Tell me more about these repair mechanisms.
Well, one of the most basic is proofreading. The DNA polymerase, that's the enzyme that copies DNA. It actually checks its work as it goes, fixes errors right away.
It's like having spell check for our DNA. That's handy.
Right. But sometimes things slip through. That's where mismatch repair comes in. It catches errors that proofreading missed.
It's like a second set of eyes going over a document.
Exactly. And it's super important. Problems with mismatch repair are linked to certain cancers, like
Yeah.
colon cancer. So it's not just about fixing typos, it's about preventing bigger problems down the line.
Wow, it's like our cells have a whole quality control department working in there. There are even more repair mechanisms, right? The source mentioned post-replication repair and excision repair.
You got it. Post-replication repair is like calling in a specialist when the regular repair crew can't handle it. And excision repair comes in two flavors: base excision and nucleotide excision.
So many options.
Think of base excision as fixing a single typo, while nucleotide excision is for bigger messes, like those caused by UV damage.
Oh, right. Like with that xeroderma pigmentosum condition, people with that are super sensitive to sunlight.
Exactly. They can't do nucleotide excision repair properly, so they're really vulnerable to UV damage and skin cancer.
That's scary. And there's even a way to fix double-strand breaks, right? When the DNA molecule completely snaps in two.
That's the most serious kind of damage for sure. We've got two main ways to tackle that: homologous recombination and non-homologous end joining.
Homologous recombination, that sounds complicated.
It is. But basically, it uses a backup copy of the chromosome to guide the repair. It's like having a spare part for your car.
And non-homologous end joining.
That's more like using duct tape. You just jam the broken ends back together. It's error-prone, but sometimes it's all you got.
Wow, this is mind-blowing. It's like a constant battle going on inside us, with all these little repair crews working overtime.
It really is. And it highlights how important these mechanisms are. Without them, we'd be in big trouble. Mutations would build up, leading to all sorts of problems, including cancer.
It's incredible how much is happening at the microscopic level to keep us healthy. This has been a fantastic overview. So now that we understand the basics of mutations, where do we go from here?
Well, next, let's look at how these tiny changes in our DNA play out on a grander scale. We'll explore their role in evolution and how they shape the diversity of life on Earth.
Sounds fascinating. I'm ready to dive deeper. We've been talking about these gene mutations, like what they are, how they happen, and even how our bodies try to fix them.
It's a lot going on in there, that's for sure.
Right. But I keep thinking, okay, so these tiny changes are happening all the time, but what does it mean in the big picture? Like for evolution?
Oh, that's where it gets really interesting. See, mutations are random, right?
Yeah.
But the environment, that's what decides which ones actually stick around.
So like a mutation could be bad in one situation, but good in another.
Exactly. Imagine you've got a bunch of organisms, right? And they've all got different mutations. Some might make them, I don't know, resistant to a disease. Others might change their camouflage.
Okay.
Now, say a disease hits that population. The ones with the resistance, they're more likely to survive, right?
Yeah, makes
Because they
sense.
reproduce, pass that mutation on. Suddenly, that mutation, it's more common. But change the environment. Say, make it so
Ah.
good, camouflage is key. Now, a different mutation becomes the winner.
It's like the environment is filtering out the mutations, keeping the ones that fit best.
Perfect analogy. That's basically natural selection at work. Random mutations happen, but the environment decides who wins the lottery, so to speak. And that's how we get all this amazing diversity of life.
The source talked about thalassemia
Right.
as an example of this whole mutation thing. What was that about again?
So thalassemia, it's a blood disorder, affects how your body makes hemoglobin. You know, the stuff in red blood cells that carries oxygen?
Yeah, yeah. And it's caused by mutations, right?
Yep, in the HBB gene. But here's the thing. There are tons of different mutations that can cause thalassemia.
Oh, so it's not like one specific typo then.
Nope. It's like multiple ways to mess up that gene. And some mutations, they cause a mild form of the disease, while others are much worse.
And the source said where you live matters too, right? Like, which mutations are common?
That's right. Certain mutations are more frequent in certain places or populations, like the sickle cell anemia mutation. It's bad news in most cases, but in areas with malaria, it actually helps people survive.
Wow. So the same mutation can be good or bad depending on where you are. It's all about context. Speaking of multiple mutations for one disease, what were those expanding DNA repeats?
Oh, those are wild. It's when a certain sequence in the DNA, it gets repeated over and over.
Mm.
And the problem is, these repeats, they can get longer over generations.
Like a stutter in the DNA.
Yeah, exactly. And as they get longer, they can mess up how genes work.
So are these repeats always bad?
Well, they're linked to some pretty serious conditions. Fragile X syndrome, myotonic dystrophy, Huntington's disease. These are all caused by these repeats getting too long, disrupting things.
It's kind of scary to think that these little repeats can have such a big impact. Makes you realize how fragile our DNA can be.
It does. But on a brighter note, remember all those awesome DNA repair mechanisms we talked about?
Oh, yeah, those are pretty cool. Our cells have ways to fix this stuff. How many different repair pathways are there anyway?
Well, it's not just one fix for everything.
Yeah.
We've got a whole toolbox. Different pathways for different types of damage, like specialized mechanics for a car, you know?
Right. We've got proofreaders, mismatch repair, post-replication repair.
Excision repair, double-strand break repair. It's a whole team effort to keep that DNA in good shape. And it's important work. Scientists even got a Nobel Prize for figuring out how some of these repairs work.
Oh, no.
Yep, back in 2015. Their research helped us understand these amazing processes that protect us from DNA damage.
It's incredible to think about all this happening inside us all the time, keeping our DNA from falling apart.
It's crucial, really, because DNA damage, it's happening constantly, right? From the
Yeah.
environment, even just our own metabolism can produce stuff that damages DNA.
So without these repair mechanisms,
Mutations would pile up, we'd be in trouble, cancer risk goes up, all sorts of problems.
it's like our health depends on these microscopic repair crews doing their jobs.
Exactly. And remember, xeroderma pigmentosum, those folks can't fix UV damage properly, so they're super sensitive to the sun.
Yeah, that's rough.
It shows how important these repair pathways are and why we need to keep studying them.
Makes you wonder if we could even learn to make them better, right?
Man,
Boost our natural defenses.
now that's an exciting thought. It's like a constant arms race, you know? Damage versus repair. And the outcome, it affects our health, our evolution, everything.
This whole DNA repair thing is fascinating. Before we move on, though, can we circle back to the Ames test?
Oh yeah, that clever little test.
It really caught my attention. Can you remind me how it works again?
Sure. So they use bacteria that can't make a certain amino acid, histidine.
Okay.
But if a chemical causes mutations, it can sometimes make those bacteria start producing histidine again.
So by watching the bacteria, we can tell if the chemical is mutagenic.
Exactly. It's like a warning system.
Mm.
And it's helped us identify a lot of nasty stuff, you know, things that cause cancer. Helps us make sure those chemicals are regulated and kept safe.
It's like a canary in the coal mine for mutagens. This deep dive into gene mutations, it's been really eye-opening. So much going on we never even think about.
It really makes you appreciate the complexity of life, right? These tiny changes, they drive evolution, they cause disease. It's all connected.
And we've just scratched the surface, I bet. There's still so much to discover.
Oh, absolutely. But before we get carried away, let's bring it back to the listener, you know. What does all this mean for us, for the future?
Great idea. Let's wrap things up by exploring the broader implications of what we've learned today. We've been on quite the journey, huh? Exploring this world of gene mutation.
It's a fascinating world, that's for sure. From tiny DNA changes to big impacts on evolution and disease. It makes you think, doesn't it?
It really does. Like, we talk about DNA all the time, but it's easy to forget how incredible it is This ancient code copied over and over, holding the instructions for life.
And yet it's not perfect, right? Those little errors, those mutations, they can have such a profound effect.
It's humbling, to think that something so small can change the course of a life. Whether it's something like eye color or a serious genetic disorder, mutations are part of what makes each of us unique.
We're all walking around with slightly different versions of the human blueprint. And those differences, they come from both random events and the long history of evolution.
It's mind-blowing. And thinking about it, one of the most exciting things about understanding mutations is how it's changing medicine.
Oh, absolutely. We're moving away from that one-size-fits-all approach. Now we can identify specific mutations that cause diseases, and that opens up a whole new world of possibilities.
Like targeted therapies, personalized medicine.
Exactly. Imagine knowing the exact mutation causing a patient's cystic fibrosis. You could tailor their treatment to that specific genetic profile, making it much more effective.
And what about gene editing? Like CRISPR, that's the one everyone's talking about. It's like we can actually go in and fix the typos in the DNA code, right?
It's incredible, isn't it? CRISPR has the potential to cure genetic diseases at their root. It's a powerful tool, but of course, with great power comes great responsibility.
Yeah, the ethical implications are huge. We have to be careful with this stuff.
Absolutely. We need to have those tough conversations, figure out where the line is.
Yeah.
But the potential benefits for treating genetic diseases, they're hard to ignore.
It's a delicate balance for sure, but it all comes back to those mutations, right? Understanding them is key to unlocking new ways to treat and prevent diseases.
Couldn't have said it better myself. So as we wrap up this deep dive into the world of gene mutation, what's the one thing you want our listeners to remember?
Well, I hope they come away with a sense of awe, at the complexity of life, at how much is happening at the microscopic level that we never even think about.
It's truly amazing. Our DNA is constantly under attack, constantly being repaired. Mutations can be both beautiful and tragic, but they're a testament to the power of evolution and the resilience of life itself.
It's been a fantastic journey, and I want to thank you for sharing your expertise with us today. It's been an honor to learn from you.
The pleasure was all mine. It's always exciting to talk about this stuff, to share the wonder of genetics with others.
And to our listeners, thank you for joining us on the deep dive. We hope you've enjoyed this exploration of gene mutation.
Keep those minds curious, folks. The world of science is full of amazing discoveries waiting to be made.
This has been The Deep Dive, your shortcut to being well-informed. We'll see you next time.